Hello everyone, welcome to this Emqn guest webinar, the first we have presented in the field of POCT, which is very exciting for us. Firstly, I'd like to thank you all for joining and to thank John for giving us time to present to us. I'll just quickly run through some housekeeping before we get started. Please note, this webinar is being recorded and will be circulated to all registrants in addition to being uploaded to the Emqn YouTube channel. The views expressed in this webinar are those of the author and do not necessarily reflect the views of Emqn. Once the webinar has finished, we'll have a short Q&A session. Please feel free to ask a question via the Q&A button at any time throughout the webinar, and we'll get to as many as possible once John has finished speaking. I'll now hand over to Dr. Rosa Mora to give a brief background about Emqn and to introduce today's speaker.
Thanks, Harry. Good afternoon, everyone. Before introducing our speaker, allow me to briefly introduce Emqn. We are an international organization dedicated to providing external quality assessment to laboratories performing diagnostic genetic tests. We offer more than 60 QA schemes to laboratories around the world. Our mission is to ensure that every diagnostic genetic test is performed with accuracy, reliability, and comparability. Emqn is not just an EQA provider, but a trust partner for collaboration. Indeed, since last year, we have been actively involved in the DEVOTE program, funded by Innovate UK. And through this program, we have worked closely with academics, clinicians like John, and industrial partners such as the GeneDrive to facilitate the implementation of point-of-care testing devices in clinical setting. We are very pleased to announce that we have successfully developed suitable EQA material for both mtRNA1 and CP2C19 point-of-care testing, which can also be used as verification material for devices to facilitate their implementation. It's now my great pleasure to introduce our esteemed guest speaker, Dr. John McDermott. John is a clinical geneticist and NIHR Doctoral Research Fellow at the University of Manchester. In 2016, he was appointed as the UK first academic clinical fellow in genomic medicine. His research focuses on applying genetic and genomic technologies to both acute and everyday healthcare settings. And notably, he secured funding for the National Institute for Health Research to lead the world first trial of a point-of-care genetic test in acute setting. With that, I will now hand over to John. John, the floor is all yours.
Thank you very much. And I'm delighted to be here today to talk about rapid point-of-care genetic testing, which is an area that our group has focused on for a number of years now. And we think is a really interesting and potential huge area of growth in the field of human genetics. So before I begin, just a few disclosures. I'm a co-founder of Father Health Limited, a health technology consultancy. I lead a range of different programs, one of which is an innovative funded technology program, DEVOTE, which involves several industry partners. I have no personal financial relationship with any of these organizations, and I've received grant funding from a range of organizations that you can see down here, but all the opinions expressed in this talk are my own. So my work focuses on the field of pharmacogenetics or pharmacogenomics, which is defined as the study of variation in DNA sequence as related to drug response. And as a clinician, when I give a medicine to a patient, I want them to have an appropriate response to that medicine, and I don't want them to have any side effects. I don't want them to have an adverse drug reaction. But we know that sometimes you give medicines to people and they have adverse drug reactions, and sometimes you give medicines to people and they have a poor response to that medicine. And put extremely simply, what we do in pharmacogenetics is that we want to increase the number of people in the population in that middle group having a good response and no adverse drug reactions, and decrease the number of individuals in the groups on either side. And clearly, on an individual level, that's really good. You don't want to have side effects. You want to respond to your medicines. But on a population level, that could have a major impact. And we know that poor medicines effectiveness and adverse drug reactions are common and costly phenomena. And they're related to a number of factors. It's not just genetics. We know that poor medicines adherence, polypharmacy, individual factors all play a role. But predisposing genetic variation is really important, and that's been recognized over the last few decades. So many medicines, before they can work, need to be activated by enzymes or absorbed by transporters. And large biobank studies, including ones that we've completed ourselves, have shown that the vast majority of us carry genetic changes which might influence the activity or the effectiveness of those enzymes or transporters. And so if you carry a genetic change that influences the activity of a certain enzyme, you might have reduced levels of an active metabolite, so reduced effectiveness of that medicine. And despite extensive evidence for multiple gene drug pairs, as I said, which have been built up over the past few decades, there remains a clear translational gap, and especially in the NHS. There's 440 gene drug pairs with prescribing evidence, and 26 of those have the highest level of prescribing evidence as defined by groups like the Clinical Pharmacogenetics Implementation Consortium, which we'll be talking about a little bit later. And so many centres around the world are putting these into practice as part of routine use. But in the UK, only 4 of those have really reached clinical practice in a routine way, although in recent years, we've seen an uptick and we're set to see more gene drug pairs implemented. So there is a clear translational gap, and that's really what my work, what my group's work specifically focuses on. And again, just to kind of reiterate, we know that addressing this is really important for individuals, but on a population level, it's going to be incredibly impactful. And that's why we've seen governments, health systems around the world particularly focus on this area. We know that adverse drug reactions just alone account for 6.5% of all hospital admissions. They take up 8,000 beds at any one time. And this is slightly outdated data now, but it's estimated that around £1.6 billion is spent by the NHS on treating adverse drug reactions annually. And that's estimated to be over 2 billion now. So addressing this poor response and adverse drug reactions is clearly really important. And so this genetic variation, as I said before, is very common, but also the medicines that it's related to is incredibly common as well. Medicines like antidepressants, antiplatelets, and pain relief. And here are the figures taken from the NHS Business Service Authority that the NHS spends on treating conditions related to these medicines. So again, I'm a clinical geneticist. I typically see patients who have rare disease or rare cancer predisposition disorders. This is not rare disease. This is common genetic variation that could impact the entire population. And so there's a clear imperative for healthcare systems around the world to think about how can we harness this and utilize it to improve patient outcomes. And so it really, to do this in practice, which is the heart of what we think about, requires a multidisciplinary and multi-stakeholder approach. As I said, we know genetic variation is incredibly common in the population. The question is, how do we take that knowledge and apply it in practice to undertake what you might call personalized medicine? And that first step is establishing what we call the genotype to phenotype relationship. So how does a change in a gene influence your response to a medicine? And once you've done that, you might have a really robust genotype phenotype relationship between a particular gene and a particular medicine, but then you need technological solutions to ensure you can access that data at the point in prescribing. It's all good and well having that data in a fancy laboratory like the one I work in, but unless you can get it into the hands of the GP or the stroke physician at the point of prescribing, it's essentially worthless. And then you may have developed a really great piece of technology, but you need to implement that in clinical practice to show that it's not going to disrupt standards of care. Genetics does not normally, historically, it's not informed routine practice in most areas of healthcare. So if we're going to start implementing genetics in a very routine way, we need to show we can do that safely, we can do that equitably, and we can do that cost effectively. And then embedded with all that, we need to ensure that this is acceptable to patients and the public, and we need to show, as I've mentioned, that it is justifiable from a health economic perspective. And one thing I would add to this is we need to ensure that the quality is there as well. And that's where quality assurance providers like Emqn definitely come into this. And we've been really good at establishing genotype to phenotype relationships in the context of rare disease and cancer predisposition disorders. That is our bread and butter. We've done that for many decades. What we've not been so good at as a clinical genetics service, but also as a health service generally, is thinking about this right side of the translational spectrum. How do we get that into practice? And that's what we in our group spend our time thinking about. The technology, how you embed that within a clinical service, and how you measure those potential improvements in outcome. So how do we do this? How do we actually get this into practice? And very broadly, again, this is kind of a lecture in and of itself, but there's three main approaches to delivering pharmacogenetics in clinical practice. One of those is what we call reactive testing, which is really what we do at the moment. A patient presents, you think they need a pharmacogenetic test. So for things like DPYD around certain cancer agents, or TPMT around azathioprine, or MTRNR1, which we're going to speak around aminoglycosides, you would send off that particular test. It would go to a laboratory. You get the results a few weeks later. That's what we do at the moment, relatively straightforward, and can be assessed using typical outcome measures. And then you have what's called reactive with planned reuse, where you're not just looking at a single gene, but you're looking at multiple genes at the same time. So you do a panel-based test. And then the idea is you can then reuse that data over a patient's life. It's more challenging to assess those outcomes, and there's increasing complexity. And then the kind of logical endpoint of all of that is preemptive testing, where you would test a large panel or potentially whole genome data at a set time in someone's life, not related to a specific medicine, but then that data is embedded within their healthcare record, and you can use that every time they present. Now, a variant of reactive testing, and one that's becoming increasingly attractive, is point of care testing. And the reason this is becoming particularly attractive is because traditional reactive testing, which, as I mentioned, has a huge number of advantages in that it's relatively simple, relatively straightforward, it is massively limited by turnaround time. So a patient presents, I think they need a pharmacogenetic test. I request that test, send it off to a centralized laboratory. We've only got a few places in the country that can do this pharmacogenetic testing, and I get the results a few weeks later, depending on how optimized the pathway is. Now, that's all good and well for certain clinical scenarios, but in many acute settings, that will just not work in clinical practice. If I said to many of my colleagues in the acute medical unit or emergency department, oh, just wait for this result, it'll take a few weeks, well, that patient will be discharged, they may have deteriorated. You need to understand how you're going to optimize prescription at that point in time. And that's really where point of care technologies, new advancing technologies, come into play. And so by doing this, by testing a patient at the bedside using a rapid genotyping approach, you circumnavigate that need to send results off to samples off to a centralized laboratory, and you overcome that issue of turnaround time. And the first example of when we really thought about this, and I know that Professor Bill Newman, who is a leader of our program here in Manchester, spoke about this recently, is around the pharmacogenetics to avoid the loss of hearing trial, which we undertook with Gene Drive in Manchester. And so aminoglycosides are commonly used around the world. They were initially identified by Selman Waxman in the 1950s, who identified streptomycin. And since then, a large number of synthetic and semi-synthetic agents have been identified. The most commonly used of these is called gentamicin, and that's used widely to treat infection, and specifically in the context of neonatal infection, which we'll come to. Now, just to take a kind of step back and think about how these aminoglycosides work. So we know that they're kind of commonly used, but what's also kind of what we're aware of is that certain individuals in certain families have a predisposition to aminoglycoside induced ototoxicity. So if I were to give any of you a large dose of aminoglycoside over a prolonged period of time, you'd likely develop an element of hearing loss. That's well understood. But it was recognized in the latter part of the 20th century that certain individuals in certain families, as I'm showing here on the screen, had a predisposition to this reaction, whereby just a single dose could cause this profound hearing loss. And what these pedigrees show, this is what we draw in clinical genetics, is that these individuals appeared to have this predisposition, and it was inherited in what we call an extranuclear or a mitochondrial inheritance fashion. And it was in 1993 that a paper came out showing that all these individuals carried the same genetic change. And this was what predisposed them to this, what we call aminoglycoside-induced ototoxicity or hearing loss. And just to kind of take a step back and think about how aminoglycosides work, this is a bacterial cell, and bacterial cells contain this 30S ribosomal subunit and then this 16S subunit. Now, when I give aminoglycosides to a person, what's actually happening is that hopefully the aminoglycosides are binding to this 16S subunit. They stop the production of proteins, they lead to truncated proteins, they cause cell stress and ultimately cell death. Now, our cells, human cells, don't contain a 16S subunit. They contain this 12S subunit, and aminoglycosides can't readily bind to this. This 12S subunit is encoded for by the RNR1 gene, and at position 1555, in the majority of us, there's an A nucleotide that's present in 499 out of 500 individuals. But in the individuals in these families who are predisposed to aminoglycoside-induced ototoxicity, there's a single nucleotide polymorphism or a mutation, as it's commonly known, at that position. So instead of having an A nucleotide, you've got a G nucleotide. This changes the conformation of the 16S subunit, so it looks of the 12S subunit, so it looks more like the 16S subunit. And so when you give aminoglycosides to these individuals in these families, aminoglycosides can bind and it causes damage and toxicity to the cell. And for a reason that's not well understood, this has a particularly profound effect on the inner ear hair cell. So this has been known about for over 3 decades now, but for a long time, we couldn't do a huge amount about it. And that's because of this issue around turnaround time. So in many centers around the world for the past decade, when a child has been diagnosed with cystic fibrosis, because it's known that they're likely to receive gentamicin at some point in their life, they were tested prospectively for this particular genetic change. And that was fine because you could send the sample off to the laboratory when they were well. And if they carried the variant, you could code it as an allergy in their records. That would work because they only needed antibiotics months or years later. And so you had no pressure, you had no time pressure. But in many clinical contexts, and this one specifically, that time pressure is very much there. So in the context of neonatal sepsis, the National Institute for Health and Care Excellence in the United Kingdom recommend that any baby should be treated with a combination of a beta-lactam, so a penicillin-based antibiotic, and an aminoglycoside. And the challenge here is you have to give antibiotics within an hour. So you just can't use laboratory-based testing. And this is a problem at scale. There's around 100,000 admissions to NICU each year based on the prevalence of the variant. That's 200 babies who are at risk. And for many years, we just accepted that that was the risk we took because we couldn't do genetic testing. So several years ago, partnering with Gene Drive based in Manchester, we asked, could we develop a technology that could deliver a clinically relevant result in a clinically relevant time frame that was point of care, cost effective, and acceptable to stakeholders? And that's what we did. We developed a 2-stage testing process, again, with Gene Drive, combining LAMP PCR with MELT curve analysis. And essentially, we created probes that were perfectly bound to the mutant allele, imperfectly bound to the wild type allele. And what that means is as you heat up that reaction as part of that kind of melting process, you get detachment and fluorescence of the wild type probe earlier, detachment and fluorescence of the mutant probe later. And what that looks like in practice to the actual system is that you see differences in the relative peak heights or positions of the peak, depending on whether you've got a mutant allele or a wild type allele. Now, the key thing here was that all this was packaged into a handheld point of care device that could test from a cheek swab from the baby, put into the system, and it could identify a result comparable to gold standard in 25 minutes. Now, potentially that was hugely impactful, and you've got a technology that all of a sudden can influence prescribing practice in that very acute setting. But clearly what we need to do, as I mentioned earlier, is to show that this isn't impacting normal standards of care. And that was that final part of that translational pathway that I showed you. And that's what we did as part of the Pharmacogenetics to Avoid Loss of Hearing study, which was published in JAMA Pediatrics a couple of years ago. And essentially what we did, we gave this system to 2 large NICUs in the northwest of England, and they embedded it within part of their routine care pathway. And what we were able to show was that it demonstrated that genetic technology can be applied in the acute setting without disrupting normal clinical practice, as measured by time to antibiotic therapy. If this technology was being used and lots of people being tested, but all of a sudden time to antibiotics went up by 35 minutes, then that would not be a good test. But what we showed was that time to antibiotic therapy was equivalent to previous practice, and 3 babies avoided aminoglycoside-induced hearing loss. This was assessed as part of the NICE early value assessment, and there is a program of work ongoing at the moment to look at how that is rolled out nationally, and hopefully there should be more news on that space relatively soon. So this was the first example of a point of care genetic technology developed and applied in the acute care setting anywhere in the world. So we addressed a number of firsts. We learned a huge amount as part of that process, but what we've seen since then is a potential acceleration in the rise of point of care genetics, and that has been fueled to some extent by the SARS-CoV-2 pandemic, the COVID pandemic, which saw companies around the world pour a huge amount of money into the development of handheld diagnostics that could identify the SARS-CoV-2 virus. Now, lots of that R&D spend could quite easily be repurposed to identify human genetic variation. So the potential of what these companies have learned and how it could be applied in clinical practice is hugely exciting. And we wrote a little piece on that a few years ago in the European Society of Human Genetics, the European Journal of Human Genetics, which you can see here and read a little bit more if you're interested in that. Now, the big talking point in point of care genetics and genetics generally, or pharmacogenetics in the UK, is the recent guidance that's been published by NICE looking at CYP2C19 genotype testing to guide clopidogrel use after ischemic stroke or transient ischemic attack. And this is a landmark recommendation. There's 100,000 patients a year, over 100,000 patients a year who have a stroke or a TIA, and the recommendation from NICE was that CYP2C19 genotyping should be offered to those patients if clopidogrel is being considered, which is the vast majority of them, to guide antiplatelet therapy. So that is a huge paradigm shift in the way we think about pharmacogenetic testing and its use in the United Kingdom. Now, critically, the first line, the first caveat to that was that quality assurance processes have to be in place for point of care tests, and that's exactly where Emqn, and we've been working with Emqn to develop these EQA systems and products which are absolutely essential. Now, laboratory-based testing was recommended, and use of the gene-drive CYP2C19 ID kit point of care test was recommended when laboratory-based testing was not available. I'm going to talk a little bit more about that later on, but I want to focus just on a bit of background on why this was considered so important as a topic area by NICE. So as a way of background, clopidogrel is one of the most commonly prescribed antiplatelet medicines in the world. It is used for a range of indications. It's used to treat patients who've had heart attacks, who've got coronary artery disease, or who've undergone percutaneous coronary intervention. It's used for neurovascular indications like stroke or TIA, and it's used for vascular surgery. And we have programs ongoing with cardiologists, vascular surgeons, and stroke physicians looking at the utility of CYP2C19 genotyping. But it is not an active substance as it is in its pill form. It first needs to be absorbed by intestinal cells, and then it's transported to the liver, where in the P450 cytochrome system, it is absorbed and it is converted by the P450 cytochrome system. Multiple enzymes are involved in that process, but the key player is CYP2C19. And then you get your active metabolite, which can inhibit platelets, and so stop platelet aggregation, preventing or hopefully reducing the risk of secondary vascular events. So CYP2C19 is absolutely essential. Now, we talk about pharmacogenetics all the time in our group, so often it's easy to forget that actually some of the language we use when talking about CYP2C19 genetic changes can be quite confusing. And I recognize there's a lot of point of care colleagues on this call today. And so I wanted to put in a few slides just explaining a little bit on the nomenclature and actually what it is we're talking about when we talk about these results, because I think that's really important. So just as an example here, when I get a result typically as a clinical geneticist, and this is an example of one of many systems that we have in our laboratory, the Agena system, but we could have an Illumina system or a Thermo Fisher system. That data is typically sent to my colleagues in bioinformatics who will do interpretation. They'll get the raw variant data and then they'll produce a report. So this is a clinical exome analysis. It's a kind of mocked up result. I've ordered testing on this patient because they've had biventricular megaly detected on antenatal scanning, and I found that they have a homozygous pathogenic variant in AMPD2. And so what you can see here is a long list of numbers and letters which are quite meaningless unless you're used to looking at these types of reports. And the reason I'm showing this is to make the point that if we're talking about pharmacogenetics as an intervention at scale that is almost ubiquitous across the healthcare system, as many people are talking about, we cannot issue reports like this. If we do, no one will use it for a number of reasons. And so we need a better way to talk about pharmacogenetic language. And this is where the star allele format arose from. So what you can see here is the raw variation, if you will, in the CYP2C19 gene. And as I said, CYP2C19 is kind of one of these key players in the P450 cytochrome system. And you can see here that you've got two copies of the CYP2C19 gene. You've got two alleles, one from mom, one from dad. And the testing has identified the 636 G2A variant and the 1297 C to T variant. Again, very confusing, doesn't kind of mean a huge amount. But then we start to enter into this world of the star allele format. And what you can see here is that you have your star alleles that are denoted by these different major variants. So here, what you can hopefully see is that because this patient carries a 636 variant and the 1297 variant, what they actually are is star 3, star 5. So that just becomes a little bit easier to talk about. So when we're talking about star alleles, what we're actually talking about underpinning that is raw genetic variation, like we would talk about any other genetic variants. So that's really important. Now, what you can do, this kind of additional step that we have in pharmacogenetics that doesn't exist elsewhere in genetics, is going into what we call metabolizer state. So groups like CPIC, as I mentioned before, the Clinical Pharmacogenetics Implementation Consortium, make these guidelines for how you combine your different star alleles in combination to get your metabolizer state. And so what you can see here, this patient star 3, star 5, they are a poor metabolizer. So again, that kind of maybe starts to mean something to clinicians in clinical practice who are thinking about putting this into practice. Now, again, probably to most people, it still won't mean anything. So then you get to the final step of this interpretive pathway. And this is the guidance that's produced by these groups. And so what you can see down here is that for CYP2C19, if you're a poor metabolizer, you should avoid clopidogrel use if possible and use an alternative agent. So if you think what we're really doing in the world of pharmacogenetics is going through from this raw variation through to actionable guidance. So patient comes in, they need clopidogrel. We go through from that raw variation to your haplotype, to your diplotype, to your phenotype, and then to your guidance. And to my mind, actually, what we need to be doing as part of the technology we're developing is presenting clinicians with either that phenotype or that actionable guidance and thinking about how we get that to clinicians in a clinically relevant format and a clinically relevant time frame. That is exactly where point of care systems come in, because especially in the context of ischemic stroke, you need that result relatively quickly for a number of reasons, not just because in some scenarios you want to get the patient on the right medicine immediately, but also because the laboratory testing is centralized. Patients might not come into a hospital where labs are available, so then you've got to send that sample to a different lab, by which point the patients come back, they might have moved to a different center. You're introducing huge amounts of complexity. Whereas if you can produce the results by the bedside, have that in their record, that is a major, major win. And there are 2 point-of-care CYP2C19s that are commercially available, or at least 2 that were considered via the NICE assessment. There's the GeneDrive CYP2C19 ID kit, which we have been part of the development process of here in Manchester as part of a number of grants over the years. And then there's the Genomics Cube, which is previously known as the Spartan test. And the Spartan test, just to say, has been assessed rigorously around the world in a number of different publications. There's lots of really good work that has used that in clinical practice. The reason the CYP2C19 the GeneDrive kit was recommended is because it covers a broader range of alleles, which is important for ethnic minority populations and individuals from different biogeographical groups where these variants are more common, specifically that STAR35 allele. And critically, there's no cold chain requirement. You don't need to put your laboratory reagents in a fridge or have a freezer storage available. And there's that bidirectional communication. So once you get the result, it can just go into the electronic healthcare record if you have that middleware integration established, which wasn't the same for the Genomics system. So the GeneDrive system was recommended, but what it was noted was that actually there was not a huge amount of work that had been published demonstrated its analytical validity. And I'm pleased to be able to present a program of work that we've undertaken as part of the DEVOTE study, which is currently under peer review. So I'm going to share that data now. Happy to share it on this call, but obviously please don't circulate beyond this call. So the system itself works by taking buccal swabs from the patient. It's then mixed in a buffer solution, and then you have a transfer pipette to move that into one of 3 tubes within a cartridge. That cartridge is then inserted into the GeneDrive system itself. You press go, and then 6 to 9 minutes later, you have a result. And essentially, using that Melt curve reaction, as I showed you before, but for the RNR1 test, essentially what you can see here, panel A shows all theoretical possible results across the 3 different wells, and what you see in panel B is for tube number 1, tube number 2, tube number 3, and they're detecting the different alleles. So depending on the peak you get or don't get in each of these tubes allows the system to algorithmically call the combination of alleles and therefore infer the metabolizer state. That's all very clever, but to the clinicians, what we're kind of simple people, what we actually see is this result. So you don't need to look at that Melt curve analysis. You see this patient is STAR1, STAR3, they're an intermediate metabolizer, and based on the current NICE guidance, they should receive an alternative antiplatelet agent. We did an independent validation of this system as part of the IPTip trial here in Manchester, and we recruited 204 individuals to this study. And we then tested them against a clinical reference test, so against the GENA system, and then where there was any discordance, we used a Sanger system as our gold standard. And what we were able to demonstrate was that compared to Sanger, the system had a sensitivity and specificity of 100% with the confidence intervals as shown here. What was really interesting is that obviously the system showed that the GeneDrive system had a high diagnostic accuracy for clinically relevant CYP2C19 variants, but it actually had a superior performance to the initial reference test that we used with a higher accuracy and a lower failure rate, and critically covered that STAR35 allele, which many laboratory systems don't. As I said, this is currently under peer review, and we should expect it to be published over the next few months. So we were incredibly impressed by the system, the way it was used in clinical practice, and if any sites are looking to implement this in clinical practice and would like to look at these results, we are happy to share them pre-publication, and we're happy to have a discussion about that, because we understand that hospitals, health systems need to feel that they have assurance over the way these systems are used in clinical practice. So what I want to move on to just as we finish, and hopefully this will kind of precipitate a bit more of a discussion that we can have at the end, is just around how rapid genetic testing really represents an entirely new paradigm, not just for us as the clinical genetics community, but also for many of you on this call as point of care providers, point of care experts. And this broad question of how do you verify and maintain a rapid point of care genetic test to ISO 15189 standards, let's say, that hasn't really been thought about a huge amount before. And I think there's some challenges there, but there's huge opportunities as a community for us to address these challenges. And I've tried to break them down into a few separate questions. I want to make clear at this stage, I am a clinical geneticist. I am not a kind of laboratory point of care expert. And so this is a starting point to enter into a discussion to hopefully try to find, to raise some questions and to hopefully try to find some answers about how do we make this easier for everyone and how do we create a streamlined and standardized approach. So there's the method and extent of verification. There's how do we source reference material, and then what does a QA process look like for this technology, which, as we know, for the NICE guidance, it is really important and just critically important to any type of genetic test. So with regard to the method and extent of verification, so I work in the Manchester Centre for Genomic Medicine, and attached to that is the Northwest NHS Genomic Laboratory Hub, and their bread and butter really is, or a huge part of their work is verifying new genetic technologies. But obviously that's in a laboratory setting. But they do that around the context of rare variants. And I think there's a lot to learn from those practices and potentially can be adapted to the implementation of point of care technology. So these are just some general thoughts. It's not kind of law by any stretch of the imagination, but thinking about how we do this in clinical practice, and if you guys were to get one of these systems, what do you need to do once it reaches your point of care laboratory and your clinical services? And the aim as part of that local verification process clearly cannot be to revalidate the technology to the same standards that we have just done as part of the DEVOTE study. That would require hundreds and hundreds of samples. It's already been done by the company and would cost tens of thousands of pounds, because what we're thinking about here is tests that, although they're cheaper than laboratory tests, they're still not the same as potentially doing a glucose test, for example. So the ISO 15189 standards state that the laboratory shall ensure the extent of the validation of examination method is sufficient to ensure the validity of results pertinent to the clinical decision making, which is kind of helpfully vague. And there's lots of different interpretations that can be made of that. And some of the thoughts that we've been having as part of attempts to try to, with point of care colleagues, to develop a standardized approach to verification is the kind of the background is that the genetic point of care tests that we're talking about here are genotyping based, meaning that the results they produced are binary. It really is a certain SNP, a single nucleotide polymorphism, or a certain diplotype, if you will, present, yes or no. It's not a result across a continuous spectrum as you might have for, let's say, a glucose reading, for example. And as such, the results can only ever be a predefined combination of those alleles. So it is either star 1, star 2, or it is not that. It is something else, but it is, yes, it is that, or is it not that? And the importance of stating that seems like a very obvious thing to say, but mathematically, compared to testing a system with a continuous output, it makes verification less onerous around the number of results, the number of samples that you potentially need to present. And we're working with a statistics team, point of care colleagues, and as part of a trial that we've just had funded, looking at how can we develop a white paper around this particular space. And we'd be really keen to work with any colleagues in this call who have ideas, opinions on how can we make this work for everyone. Because the idea that every point of care team would need to develop their own local verification protocol seems onerous and inappropriate. So for several centres in England, we provided 10 buccal samples of varying diplotypes to be tested in the laboratory, then undertaken gold standard genotyping for the first 15 samples collected in clinic. And that's the approach we've taken, and a number of point of care colleagues have also taken. But as I said, formal standardization of this approach is urgently needed. It's completely unfair on point of care colleagues that they're having to interpret this and make up their own guidance, or develop their own guidance each and every time a system is delivered. So at least if there was some structure or some framework around that, my sense is that would be incredibly beneficial. But obviously, to do that, you need to have reference material, and Emqn have introduced the webinar today and have kindly asked me to talk. And we've worked with them as part of the Devote program to develop some of that reference material and look at how we can develop a QA process. So in the short term, if let's say over the next few weeks, you guys say, well, we want to implement this in clinical practice, we have ethical consent to share anonymized buccal samples of a number of different diplotypes to support the verification of these systems. But clearly, that's not a scalable solution, and longer term support provided by partners like Emqn will be absolutely essential. And when it comes to a QA process, most point of care technology providers, including GeneDrive, provide their own control material to support with regular QA of the systems, typically every 2 weeks. But again, Emqn have developed a really interesting patient-derived cell-based swab system. So rather than just getting a vial of DNA or a buccal sample, what you're actually getting is a swab with cells adhered to it, which then you can put in to the buffer solution, and you are replicating far more of that end-to-end process. It's a really neat solution. It's been really interesting to see that developed, and I think it is something that could add real value, not just to the point of care community, but also the genetics community as a whole. And I know that Emqn are launching a pilot EQA scheme for mTRNR1 testing in early October 24 and CYP2C19 testing early next year, addressing those issues in the NICE guidance. And just to kind of finish up, why have NICE really addressed this and made this recommendation? I mean, clearly patient benefit is key, is fundamental, but just to go back to why pharmacogenetics is garnering so much interest around the world. So we undertook a health economic assessment, much of which informed the most recent guidelines, looking at cost-effectiveness assessment of using the GeneDrive point of care system in stroke patients to guide therapy using what's called a decision analytic model, with a Markov model. And so that looks at the different potential states of health that an individual can be in after having a stroke. And what this showed is that essentially CYP2C19 point of care testing was dominant. So it suggested that point of care testing would generate a net benefit of QALYs. So patients were healthier and had an improved quality of life. And it would also generate a net monetary benefit per patient for the health system. So using a rapid point of care test was £512 less expensive per patient. And across the health system, when you include QALYs and costs saved in readmissions, for example, the savings to the NHS are significant. And so depending on sensitivity analyses, they can go up, they can go down, more rigorous assessments need to be done, which they were done as part of the NICE guidance. But essentially, pharmacogenetic testing has the potential to improve patient safety, but also save money for the healthcare system as a whole. I hope that's given you an overview of the work that we do. We really do think that point of care technology has a significant potential to impact patient care and benefit patients across the NHS, and providers like Emqn are going to be absolutely essential in supporting that delivery, ensuring that quality is robust. So I think that leaves us 15 minutes for questions. I'm very keen to take questions and talk a little bit more about our work. So thanks very much.
Thank you, John, very much for such an inspiring, informative talk. So whilst we wait for a few more questions to come in, I would like to start with one. So as you mentioned, NICE have recommended laboratory testing as first line for the CYP2C19 genotype. And I think this is from a while now. So I was wondering, in which clinical scenario do you think point of care testing is actually a valuable addition to this already well-established infrastructure?
Yeah, no, so the NICE guidance made a recommendation that laboratory testing was to be first line if available, and then if not available, to look at point of care solutions. We're undertaking a program of work looking at how those different models might be delivered in clinical practice. And what we're increasingly aware of is that there are complexities around delivering laboratory-based solutions. Now, if you're seeing a patient potentially in an outpatient clinic and you have time to get that result, then yeah, fine, use laboratory testing if you want. You can send off the samples and get the results a few weeks later, because you've then got a single point of contact for that consultant who's ordered the test, let's say. What worries me and clinical colleagues around the country, and we've spoken to a lot of them around that laboratory-based solution in the acute setting, is anyone who has worked on the kind of shop floor, as it were, and seen patients coming in, and then you order a test. In the context of stroke care, if you are seeing a patient in that acute setting and you order, let's say, a CYP2C19 test that then goes off to a different laboratory, there are challenges in, if you're sending a test out by your biochemistry lab to a different center, that's challenging and comes with cost. There then has to be a transport infrastructure from every hospital in the country to get the samples to the genomic laboratory hubs, let's say. That comes with cost. There's then the complexity on the laboratory side around how those results are returned back to the clinicians when the person who ordered the test may be a junior doctor who isn't on shift or has moved to a different site, and the patient may have moved to a different site as well. So, and even if, let's say they've been discharged, you've then got to bring them back. And all of those things come with costs. And a micro costing assessment, I'm sure, would show that the actual cost of laboratory testing is significantly higher than, as we know, than the cost of the laboratory test itself. And so if you're doing this on a relatively small scale, it can be done. And I'd say our colleagues in Dundee have done a beautiful piece of work setting up CYP2C19 testing from a laboratory perspective. That's absolutely brilliant. And I think there's huge learnings we can take. But I'm under no illusion that I think that in England, the way the system is set up, the likelihood that we are just going to be delivering laboratory testing is incredibly small. And we know our colleagues in stroke care are keen to have point of care technology if we can make it available and make it work. And it's not about the testing, it's about how we create a landscape to allow hospitals to have that from a commissioning perspective and from a quality perspective. So neither approach is perfect, but I think it's about making them work together.
Thanks, John, for such an actually comprehensive answer. I think we have a couple more questions. One actually is related to the cost, that are still a barrier, I think, to clinical implementation. So obviously, when NICE recommended one of these point of care testing, this facilitated. However, the cost is probably still a big issue. So how do you think point of care test implementation is going, actually, also outside the UK?
So just so I'm understanding the question there. So the question is around the
barrier.
The cost of point of care testing.
Yeah.
Okay.
Implementation. Yes, please.
Yeah. So I mean, the way certainly in the UK, the way our health system works is NICE take an extra welfarist view to judging whether interventions should be funded, should be used. And so we absolutely have to demonstrate that cost effectiveness analysis, and that ideally, if a test is cost dominant, that's where we want to get to. And that has to be rigorous. So it can't just take in the cost of the technology. As I said before around laboratory testing, it has to take that micro costing approach and work out, like, if you're asking a junior doctor to take 20 minutes out of their time to go and take a blood sample, work out how you send that off to a laboratory, that comes with costs as well. And so it's capturing all that information. I think what the NICE guidance has shown is that the potential savings here are so enormous that that it is worth doing for the health system, even if the initial outlay is relatively significant. I think when you're talking about pharmacogenetics generally, whether you're talking about like panel-based testing and implementation as kind of like a screening approach as part of patients' medical records, that's a different question, and one that our team are kind of independently addressing. But I think from a single gene drug perspective, the evidence around point of care testing for CYP2C19 in stroke is incredibly strong from a health economic perspective.
Thank you.
Thanks, John. The next question, I don't know how involved you were with the decision making on the design of the assay, but maybe you could answer it from a clinical point of view. Is the decision on the variants included in the gene drive kit and mainly the reasons behind why they were chosen?
Yeah, so we certainly weren't involved in the design from a kind of hands-on different primer approach. Our input was a kind of clinical one, and our input was that the variants you should try to include as many clinically relevant variants as possible that have strong or good evidence within the CPIC guidance. So if you can include so you have to include star 2, star 3, star 17 because that's the kind of tier 1 guidance. And just say that that is another way you can approach it. There is the Association of Molecular Pathology in the US that have tier 1 guidance, tier 2 guidance or tier 1 variants, tier 2 variants. So you have to include all the tier 1 variants and then essentially as many as the tier 2 variants as they could that had good evidence around it. That was our recommendation, and fair play to the company, that's what they did.
Thank you. Another question is about, do you think there are applications of point of care testing in pharmacogenetics outside of acute setting?
Yeah, so I think that what point of care allows is a kind of democratization of access to genetic testing without the need to go to a centralized laboratory. It's really about time and those costing models. So essentially, if you're seeing someone who, let's say, they're struggling with their pain relief or struggling with their antidepressants, then that you may be able to develop a model where you do a panel-based test in a laboratory and then you can get the results for the next time you see them. That is probably the appropriate model in that particular clinical scenario. Now, you may be in a healthcare setting where you don't have access to centralized great laboratories like we do in England and actually or in the UK. And if there was a point of care test available that could do a kind of relatively modest panel, then that's potentially really exciting, especially if you're in maybe a private healthcare setting or a setting where you don't have those laboratories. So I think it's about the time you need the results and the healthcare context you work in. In the UK, I think the benefit of point of care is clearly around that acute and urgent setting. I think in parallel, what we will see is more of this data embedded into patients' healthcare records as part of panel-based testing for that more chronic, non-urgent healthcare application.
Thank you.
Thanks, John. The next question, we've obviously spoken a lot about the NHS and the implementation within the UK, as that's with the system we're working in. But the question is regarding the implementation in healthcare systems with different structures, such as private healthcare. Wondering if you have any thoughts on that.
Yeah. So the technology. So again, not specifically speaking about the gene drive technology, but this technology generally should be agnostic of healthcare setting, providing you've got the correct regulations around whether it's UKCA marking, IVDR marking, FDA marking. Providing you've got that, then it has benefit in lots of different healthcare settings and systems. I think the reason that we've seen uptake in other healthcare systems is that individual organizations can potentially move quicker than the NHS in the sense of if you work at the Mayo, for example, or Vanderbilt, then maybe it's easier to leverage your budget to say, well, we want to do that at our institution, where that's somewhat harder across the entirety of the NHS. But when we do move, we move at scale, and that's really exciting. So I don't think there's anything about another healthcare system in particular that this wouldn't be valuable in. There is no healthcare system out there that has got anywhere near having preemptive pharmacogenetic testing with all this data in patients' records. Even the most advanced healthcare systems are a way off that yet. So I think point of care testing has value across different health settings and different geographies. And again, just to echo what I mentioned before, in those settings where you maybe don't have access to centralized laboratory infrastructure, that's where it could be particularly powerful.
Thank you. Another question is about what do you think will be the next point-of-care test biomarker target to come into clinic practice?
The next point of care
Biomarker.
biomarker. I mean, hard to say. I mean, lots of people have been looking at ApoE in the context of some of the new monoclonals, although the most recent NICE recommendation may slightly change that, but I know that Genomics have looked at developing an ApoE test. Again, I think it's depending on where the technology goes. Like if all of a sudden you can get to some kind of technology where you can do a panel-based test and you can even start looking at things like CYP2D6, which is metabolizes 25% of all medicines, then that's incredibly powerful. So I think for now, CYP2C19, MTRNR1, potentially DPYD, ApoE, they're the kind of key targets that you're working with. And there is a huge the question I don't think is just making more targets. It's about applying them properly in different healthcare contexts. So in the UK, again, sorry to keep going back to the UK, but we're only doing this or starting doing this around stroke. But we know that the evidence is almost more robust in cardiovascular disease, and then there's lots of evidence in vascular surgery. So I think it's applying it to different clinical contexts as opposed to just developing more markers for now.
Thank you.
Thanks,
so
John.
I think, sorry, Harry, go ahead.
The next question is compared to whole genome sequencing, which is becoming consistently cheaper. The cost of having multiple point-of-care tests over a lifetime could be more expensive. And this question is wondering, have you looked into the speed, accuracy, and cost benefit of point-of-care testing compared to a whole genome sequence?
So that direct comparison hasn't happened, although I am pretty certain that it would show that point-of-care testing was significantly more cost effective, even if you probably did 10 point-of-care tests at the moment. But then the benefit of whole genome sequencing is that in theory, you're not just getting CYP2C19, you're then getting lots of other information from it, and that's really exciting, really valuable. And I think there will be a lot we're working in that space. So I'm not poo-pooing WGS by any stretch of the imagination. But clearly you can't generate a WGS in an acute setting. So that's kind of not applicable to the acute setting if you've not already had testing. If you've already had testing and you've got that WGS available, or you've had one previously, then at the moment, there's no way to readily access that information. Certainly as, let's say, a stroke physician at a hospital, you can't just go and look at their WGS. That's impossible. It's hard for even clinical geneticists to do that. And the cost of storing WGS, the cost of that interpretation pipeline, it's really complicated. So they both have their merits, they both have the disadvantages, but I think for a long time that point of care CYP2C19 testing will be more economically viable than doing WGS in this particular context.
Thank you. I think it's now time to close this section. Thank you once again, John, for your valuable insight and for sharing your expertise with us today. It has been really an inspiring section, and we greatly appreciate the time and effort you have dedicated to discussing this exciting development in point-of-care testing. I also would like to thank you all our participants for joining today's webinar and for you engaging with the question. I think that, in the end is through this kind of collaboration and knowledge sharing that can really, help this new emerging area. Before we close, I would also like to remind everyone that a recording of the webinar will be available shortly. And if there are any remaining questions that weren't addressed today, can be submitted to the Emqn team for follow-up. We also encourage you to stay connected with us for updates on future webinars and development of point-of-care testing genomics EQA. And on behalf of Emqn and all of us here, thank you once again. We look forward to continue this important discussion in the future. Have a great afternoon, everyone. Bye-bye.